All right. Hey guys, welcome to the second part of 6.1. In this particular video, we're going to look at meiosis. So let's jump into it. All right. So meiosis, by definition, is the production of gametes, which are haploid in number. Okay. It happens in the gonads, which are your sex organs. So for males, it occurs in the germ cells that produce the sperm in the testes, and then in females, the germ cells produce the eggs inside the ovaries. Okay, and through meiosis, we go from 46 chromosomes in an adult, cell into 23 haploid inside the egg, or 23 haploid in the sperm, and then together, the egg and the sperm fertilize. The sperm fertilizes the egg, and then you get 46 in the zygote, and then mitosis then replicates that so that you have growth in the individual. Okay, so meiosis is not the opposite of mitosis. Mitosis, by definition, is division of the nucleus and cell replication, but meiosis is the formation of the gametes. But you can see there that the processes are very similar, and especially when we go through each phase, you're going to notice that the naming of the phases are pretty much the same, with the exception of a few numbers. Okay. So mitosis does not involve the recombination of alleles, and it creates daughter cells that are going to be genetically identical, right? You get two daughter cells as a result, and then the number of chromosomes produced from mitosis is going to be the same as the parent. And that makes a lot of sense. If your cells are replicating, you want to have the same number, okay? Meiosis, on the other hand, rearranges alleles between chromosome pairs, creating daughter cells that are going to be genetically unique. You want unique sperm that are going to be different in the haploid number from that of the individual parent, the dad, or the egg from the mom, okay? Unlike mitosis, meiosis also forms 4 daughter cells per parent cell as opposed to 2. And then the daughter cells produced from meiosis will have half the number of chromosomes from the parent, because half and half will make a whole new individual. Okay, here's an overview of it, and you can see there that there are 8 stages in meiosis taken in 2 rounds. The phase names are similar to mitosis, but they will always have a number next to them, and that's the telltale between mitosis and meiosis. When you see a number next to it, it is meiosis. When you see no number, it is mitosis, okay? You can basically take a look at this particular graph to kind of show you the overview, where you get interphase. Through meiosis, you get 2 round 1, and then 2 again in round 2, giving you half the amount of chromosomes you would expect in the adult. All right, let's go through each one. We're going to start with meiosis round 1. And the first phase in meiosis round 1 is prophase 1. And you can see there the number 1 indicates that it is a meiosis. Now, for the most part, it is very similar to mitosis, right? So in the nucleus, you're going to get chromatins shorten and condense to form your chromatids, and then they become visible. The chromatids then form your chromosomes. In the cytosol, your centrioles, which are the organelles that release the spindle fibers, are going to move to the edges of the cell, and the spindle fibers are going to radiate to form the spindle network, and your nuclear membrane is going to dissolve. So very classic mitosis, right? Prophase. But there's more. Here, the matching pairs of chromosomes, which are homologous, are going to line up and exchange DNA with one another. That's called synapsis, when they line up next to each other in pairs. And then what they're going to do is they're going to exchange their DNA via a process called crossing over. The gene is from the same gene loci of both chromosomes, so that you're exchanging the individual particular gene and you're recombining the alleles of the gene, right? The resulting chromosome is a recombination of the 2 previous chromosomes, and the DNA of each chromatid is going to differ from the other, right? So for example, here you've got chromosome number 1 and chromosome number 2. They're homologous, and they're going to match up next to each other. Chromosome number 1 has capital A for that particular gene, and then this one has a lowercase a, capital B, capital C, lowercase b, lowercase c, and then this is crossing over, and then they're going to detach and exchange that little segment. And so now you're going to get what was on the red chromosome going to be attached to the blue one, and the blue one attached to the red one, and you're going to get a different lineup of chromosomes from here. Now, make a note of this mental image, because it's going to reappear later, okay? Now, the only chromosome set that doesn't do this is the male sex chromosome. Why? Because the X and the Y are not homologous, and therefore it can't happen in the sex chromosomes for the X and the Y, okay? It can happen for the X and the X, but not for the X and the Y, okay? So crossing over and recombination occurs in prophase 1. Now, in metaphase 1, your chromosome is going to line up at the equator along the metaphase plate, and they're going to attach themselves to the spindle fibers, okay? The chromosomes are going to lie horizontal so that each chromatid is faces away. The nucleus by now is fully dissolved. However, the only difference that you're going to see here between metaphase 1 and metaphase in mitosis is that they're not going to line up in singular. They're going to line up in pairs, right? So metaphase in mitosis lines 23 up along the same equator plate like in singular. 46 chromosomes up along here. Whereas in metaphase 1, you're going to see 23 pairs line up next to each other in this particular fashion, okay? The positioning of the chromosomes along the equator is random, okay? This allows them to be sorted in no particular pattern, and we call that independent assortment, right? This is very advantageous for the cell because later on when they shuffle this, it's going to then give you as much diversity in your sperm as possible, or your egg as possible, okay? So that's metaphase 1. In anaphase 1, the centromere divides and it's going to pull each double-stranded chromosome to their respective sides. Your spindle fibers are going to shorten in order to pull that process, and the homologous pairs are going to move to opposite ends of the cell, and each half, unlike anaphase in mitosis, is not going to be genetically identical, right? So in mitosis, in the anaphase, you're going to have this exactly the same as this exactly the same as this, and so forth, because each half is being ripped apart. However, because they lined up in pairs in meiosis, in anaphase 1, each homologue of the pair is going to be pulled to opposite sides of the pole, and you can see they're not exactly the same. They're mostly, kind of crossed over and recombinant already, right? And then you get to telophase 1, where the two sets are now going to form tight clusters again. A new nuclear envelope is going to form, creating two new nuclei within the cell. Your spindle fiber is going to disassemble. Remember that each half no longer has identical DNA, which is the purpose of it, and the chromosomes are going to unwind and become chromatins again, so that cytokinesis can occur. The cell membrane is going to pinch off through cleavage and divide the cell back into two. And remember that for the most part, cytokinesis happens simultaneously to telophase 1, okay? So there you go, you've got your two cells splitting off slowly, and then this particular half is not going to be exactly the same as this particular half because of that crossover recombination and independent assortment process. Now, when you get to round 2, it's basically like mitosis. Everything pretty much happens like mitosis, except you're doing it with less chromosomes, okay? So in prophase 2, the chromosomes are going to condense, the nuclear envelope dissolves, and then the spindle fibers form their network again as the centrioles move to their poles. There is no crossing over in prophase 2. In metaphase 2, your nucleus is fully dissolved. The chromosomes are now going to line up singularly along the equator, and then the spindle fibers attach to the centromere. In anaphase 2, the spindle fibers are going to shorten, and then the chromatids are now going to be pulled apart from one another, just like in mitosis, and each side will have different DNA from the other side. And then in telophase 1, the chromatids are going to uncoil and become invisible, a new nuclear envelope is going to form, and you're going to get 4 haploid nuclei forming from the 2 daughter cells that have just formed after round 1. Cytokinesis is going to occur simultaneously, and now you get 4 haploid cells, okay? Now, I'm not going to go through everything that's on this particular slide, but it is a summary that's going to be very useful for you to have a look at. But in general, you can see there prophase 1, metaphase 1, anaphase 1, telophase, giving you 2 by the end of that. And then each one is going to undergo round 2 separately, and then you get 4. 1, 2, 3, 4 by the end of round 2, okay? Now, let's talk a little bit about crossover and recombination, because sometimes it can be a little bit hard to understand, right? By the end of meiosis, one diploid cell is going to produce 4 haploid cells, right? So one germ cell is going to produce 4 sperm, and that those gametes are going to be all genetically different from one another because of crossover and recombination. So you can imagine there are your homologous pairs from the one cell at the start, right? And then what you're going to get is when they cross over, right, after that particular stage, right, they're going to look, a little bit different down the bottom. But after round 2, right, at the start of round 2, when you tear them apart from one another, right, that's going to go to one cell, that's going to go to another cell, and then each half of these are going to be torn apart as well, and you end up with one that's completely blue, one that's mostly blue and a little bit red, which is this one over here, One is mostly red and a little bit blue, which is this one over here. And then this one is all red, which is over here, right? So you can see that by the end of round 2, this is going to go into a sperm, this is going to go into a sperm, this is going to go into a sperm, and this is going to go into a sperm. And that gives you 4 different variations of the same chromosome and the same set of genes, okay? Now, that happens in 2 of your chromosomes, right? You can imagine that by the time you get to 46 chromosomes or 23 pairs, that's going to be super, super different from one another, right? So this is why each sperm is going to be genetically different from each other sperm, which is why you have siblings that have the same mother and father but still look different from one another, right? Now, in terms of naming, the two ones that have crossed over are what we call recombinant chromatids, and then the 2 that haven't crossed over are what we call non-recombinant chromatids. So you can see they're black, black and green, and then green and black, and then green and green, right? The black and the green are non-recombinant because they were the same, but the ones that have recombined are called recombinant, okay? So to mathematically calculate what are the chances of 2 sperm receiving the same set of chromosomes, you can use the formula 2n equals 2, whatever the number of chromosomes is, right? So if if 2n equals to 4, the diploid number, if there are 4 chromosomes in it, you get a 1 in 16 chance. If you have 6, you have a 1 in 64. You can see as you increase in number, it becomes more sophisticated, right? The chances are just so small that they are going to be the same, right? So by the time you have, 2n equals to 46, n equals to 23, the chances are now in the millions. It's a 1 in a million chance that your 2 cells are going to have exactly the same DNA from one another, okay? Now, not only does crossover and recombination give you different genes, but independent assortment also gives you different genes as well, right? So independent assortment is when you randomly assort the paternal and the maternal chromosomes in pairs along the equator, right? So that results in independent pairing, and it's going to also produce genetically different outcomes in the gametes as well. So here's an example. Imagine if your individual has 4 chromosomes, right, in 2 homologous pairs. 1, 2, 3, 4. Now, if they was to sort themselves like this, you would get all the blues on one side, all the reds on one side, and then it splits in half in meiosis round 1, and then it splits again in meiosis round 2, and you get the first combination, which is all blue, and then the second one, which is all red. Now, the second possibility is, imagine with those same 4 chromosomes, right? Rather than being sorted blues to one side and reds to the other side, you might get an exchange, right? Blue on one side, red on the other side, and then red on the one side, and then blue on the other side. And so this particular second set here has swapped around independently, randomly, rather than being the same formation, they've swapped over, right? So, this sperm could have possibility number 1, but then this sperm might have possibility number 2. And then when you split them down the middle, you're going to get blue and red. And then on top of that, you're going to get a combination that looks something like that, right? So if you were to multiply this by 23 pairs, that's going to give you vastly different combinations of sperm again. And so once again, if you want to calculate that, the possible combination is 2 to the power of n, where n is your haploid number. So if n equals to 23, there is an 8.3 million possible outcomes you can get in all of the different sperms, right? That is just through independent assortment alone. So the chances of 2 sperms being the same because of independent assortment is going to be 1 in 8.3 million. You combine that with if you combine that with crossover and recombination, we are talking about billions. It's like literally impossible for the 2 sperms to be the same and have the exact same possibility of DNA. That's why siblings look different from one another, right? Okay, now, last thing to note. So when we talk about this production of the sperm and egg, spermatogenesis, which is sperm production, and oogenesis, which is egg production, are actually slightly different in the process of meiosis, right? For sperm, one spermatocyte produces 4 sperm cells, right? However, for eggs, one oocyte, which is your germ cell for the egg, is going to produce 1 ovum and 3 what we call polar bodies. Most of the nutrients is diverted away from the polar bodies and into the ovum. And so the polar bodies are essentially useless. They're not entirely useless, but they're essentially useless. And the ovum becomes the egg that we are referring to, okay? So there's meiosis, and then these cells here, they're going to turn into sperm, right? So they turn into sperm, and you get 4 different sperm with very, very unique DNA. However, the egg, what's going to happen is as they divide, that ovum, that's going to suck all the life out of these polar bodies and turn them into little shriveled peas. And then the ovum becomes the actual egg, which is why the ovum is so big. The egg is massive compared to the sperm. And here is a micrograph of the sperm there trying to get into the egg, and you can see the egg's massive, and then the sperm, that little thing there, that's the sperm. All it does is basically trying to bore itself into the egg and delivers its DNA, and that's pretty much it. Whereas the egg has all of that nutrients that it's going to need in order to develop and grow and go through mitosis. Okay, so that's pretty much it. Lastly, we're going to take a little bit of a look at different chromosome disorders. Okay, so what happens when the meiosis process might actually stuff up a little bit and you get different chromosome problems as a result of that? Well, depending on what the type of chromosome disorder is, you're going to get a different condition. Okay, so there are 5 different ways in which chromosomes can kind of stuff up when they are being sorted through meiosis, right? The first one's what we call deletion. By pure accident, a section of the chromosome has been removed and lost, and then the chromosome shortens and all of the genes inside that section is gone, right? You can have duplication, where a section of the chromosome is going to duplicate itself and it's going to be longer than it's supposed to be. And you might think that's pretty harmless, but cells rely on particular gene loci in order to express the genes and produce the proteins. And so when you have 2 copies, the cell struggles to read its location and find the relevant gene. So that's also going to be a problem. You can sometimes flip the chromosome segment, which is what we call an inversion. So what was at the top now goes at the bottom, and what was at the bottom goes at the top, and then it's read backwards by accident, and that's going to create problems because you create different proteins based on the backwards reading of them. 2 chromosomes can be substituted, which is when you take a section of one and you insert it into another one, or it can be translocated, where you have 2 sections that are going to exchange with one another. Different conditions are a result of different chromosomal changes. Okay, so for example, Down syndrome is a duplication of an entire chromosome, and you get 3 copies of trisomy 21, and it presents a few different problems. Klinefelter syndrome produces an extra X chromosome, so you get XXY, which is 47 chromosomes. Patau syndromes will have 3 copies of chromosome 13, and Turner syndrome is when all or part of the X chromosome is altered or missing, which is monosomy, and you have 45 complete chromosomes instead of your average ones. Okay. Now, here is Down syndrome. If you were to look at an individual with Down syndrome, they are characterized by a few different traits: mental impairment, physical growth delays, low muscle tone, increased risk of health problems such as heart defects, leukemia, and thyroid disease. And then characteristically, in a lot of Down syndrome children or people, you have this kind of double eyelid that forms just above their eyes, and then a very kind of enlarged forehead. That's because the proteins that are responsible for formation of the eyelids are actually in chromosome 21, which has been duplicated, and there are complications in the way in which they are being expressed, kind of too much almost. Here is the karyotype of this individual, and you can see there all your pairs present and 47, and you got you got 21, 21, and 21, which is why they call it trisomy, tri meaning 3, trisomy 21. Okay, this individual is a female, right? So you can see there by the extra X chromosome. All right, well, that's it from me. I hope you guys enjoyed this video, and I will see you guys in 6.2.